Day everyone. So today we'll be discussing the second part of the life cycle of Puccinia. We have already studied that Puccinia is a heterogeneous fungi and that it has two hosts. The primary host is wheat and secondary host is barberry. On the wheat plant, the binucleated acidiospores they fall on the wheat plant and they put forth germ tube. They flourish inside the tissue of the wheat plant, and then it produces uridospores, and then telutospores, and finally basidiospores. That's these things we have already discussed earlier, and now we are going to study how the basidiospores which are produced on the wheat plant are not capable of infecting the very same wheat plant. Instead, it needs a secondary host, or the haploid basidiospores need another plant for it to survive. So in this video we're going to discuss how Puccinia infects on its secondary host, that is the barberry plant. Okay, so in the wheat plant we have already discussed that all the spores are binucleated spores. Okay, but here we can see that the spore will be it will be haploid, and also it will be uninucleated. Okay, so that's the difference. So let's see, after it falls on the barberry plant, what happens. So this is the story which is continued after the basidial stage. The basidiospores, it represent the beginning of the haploid phase. The basidiospores, as you know, are incapable of infecting the wheat plant, so they fall on its alternate host, that is the barberry plant. The haploid phase, it consists of haplomycelium, and two spore stages arise. One is the pycnidial stage and the acidial stage. So the basidiospores produce two spore stages. Previously, how many spore stages we have studied? Three spore stages: the uridospores, telutospores, and basidiospores. Here we will be studying two spore stages, which are pycnidiospores and acidiospores. Within the host tissue, the basidiospore germinate by putting forth the germ tube, and it grows into form the monokaryotic mycelium. The mycelium is septate, uninucleate, and haploid. Okay. Now there are two types of hyphae are produced, that is the positive strain hyphae and the negative strain hyphae. Okay. And now what happens is, this hyphae produces pycnidial stage on the upper surface and the acidial stage on the lower surface. Now let's see the pycnidial stage. The monokaryotic mycelium establishing beneath the upper epidermis of the barberry leaf. From this mycelium, it arises a group of hyphae and forms the pycnidium. Okay, the pycnidium may be positive pycnidium or negative pycnidium, depending upon from which mycelium it was formed. Now when you look at the structure, we can see that in the above, that is in the upper epidermis, you can see that there is a tiny space is provided with a red rectangle. If you can see, we can see that there is a red rectangle, and inside the red rectangle you can see the pycnidium is marked. Okay, not the lower epidermis, it's not on the lower side, it's on the upper side, so be careful. On the upper side you can see the pycnidium. Okay, now let's discuss the structure of pycnidium. Pycnidium is a flask-shaped structure. It is a flask-shaped structure. It opens to the outside by a small pore, and the pore is called ostiole. Okay, the ostiole, it is guarded by sterile hyphae called the periphysis. Two types of hyphae are seen in the pycnidium, that is the long and delicate receptive hyphae, which is found on the outside, and short spermatial hyphae, which are found inside the flask-shaped cavity. The spermatial hyphae bears the spermatia or the spicnidiospores on its tip, and the spermatia is a non-motile or oval, okay, with a large nucleus. The pycnidium give rise to positive and negative spermatia. Now, this spermatia are transferred from one pycnidium to the other by insects, and the spermatium of one strain, that the positive strain, is transferred to the negative, the spermatia of the hyphae with the negative receptive hyphae with the negative strain. Okay, and the positive and negative spermatia, they come close to each other, they fuse together. Okay, so I hope you have understood. The pycnidium is formed on the, it is a flash shaped structure which is formed on the upper surface of the a leaf, barberry leaf, okay, on the upper surface, and it is a flash shaped structure. Remember, it is a flash shaped structure. It has a it has receptive hyphae on the towards the outside, towards the upper epidermis. You can see there is receptive hyphae. This receptive hyphae can be seen protruding from the epidermis. It can be seen projecting from the epidermis. Okay, and the spermatiophores or the pycnidiospores, they are of two types. What are they? They are the positive pycnidiospores and the negative pycnidiospores. The pycnidiospore of one strain is transferred to the other pycnidium of the other strain, like positive pycnidiospore is transferred from the positive pycnidium to the negative pycnidium through what? Through insects. Okay, and uh, and let's see what happens after that. So once the transfer is done, uh, we can see that the intervening wall between the two of the pycnidiospores, they dissolve, and the two nuclei, these are haploid nuclei, remember, the haploid nuclei come in contact with each other, and they remain as a dikaryon. That means they remain as a dinucleated structure, and this, from this dikaryotic cell, a dikaryotic mycelium is formed. Okay, this mycelium forms the ascidia. The ascidia is embedded in the mesophyll of the barberry leaf, but it opened towards the lower epidermis. They are cup shaped structure. Remember, pycnidium was flash shaped structure. This is cup shaped structure. Okay, so if you can just rewind back and go to the picture we have already uh, seen, you can see flash shaped or barrel shaped structures towards the lower epidermis, and that is the ascidiospores. Now the ascidiospores are produced in the basipetal succession. That means the lower ones mature first. These spores are liberated by disintegration of the intercalary cells. The cells which surround them, they disintegrate, and that's how these ascidiospores are liberated. Now the mature ascidiospore, it is binucleated structure, okay, and it is thick walled and globose or subglobose or polyhedral. It's not exactly global, it is subglobose, otherwise it is polyhedral in shape, with the germ pores. And these ascidiospores cannot germinate on the barberry leaf. Instead, where do they germinate? They germinate on the wheat plant. So these ascidiospores are carried by air and they are transferred to wheat plant. Okay.